Good morning, everybody. Thank you so much for joining me this morning. Hope you guys had a great weekend and can really appreciate the fact that you guys are halfway through this experience already. So good job. I know that this is a lot of information to throw at you all at the same time. As I said on the first day that we had together, this is a strenuous course even during a semester, and you guys are taking it in just a few weeks. So you guys are rock stars and definitely pushing through. I know there was a lot going on this weekend, kind of a tie-over from last week. Are there any questions, comments, or concerns that I can address at this point to the entire class? Okay, and I'm sure you guys saw that I did post grades, or at least what I was able to get through on grades. In general, the grades are actually higher than they would normally be in an in-person either spring or fall semester or even over the summer. So that is fantastic news to me, and hopefully it is to you guys as well. At this point, you know, there are only really grades for the first quarter of the semester, and so if you aren't exactly where you want to be at this point, no worries. There's still plenty of time, right? There's still 3 more exams, 2 practicals, including the one that opens today, and so there's a lot of opportunities to bring up your grade. And perhaps you guys saw the little analysis that I did of the videos and their effect on multiple choice scores, and so it does seem like there is a link between making those videos, at least, you know, studying them enough so that you can talk about it, and actually doing better on the multiple choice section. So that is interesting to me, and hopefully validates the experience. And so even if going forward we don't do those types of videos anymore, I definitely would like to point out that that is a really great study tool for you guys, even going forward into AMP2 and into your graduate programs eventually. Okay. Again, I'll pause just in case anyone has anything they want to throw out for the good of the order. Okay, and I've actually really enjoyed watching those videos. It is wonderful to see and hear you guys, like really wonderful, but also you're doing just such a phenomenal job. It is incredible that you guys can, you know, sit at home and learn these things to such a thorough degree. So you really should be proud. Okay, so this week we are wrapping up information for lecture exam 2 for, so wrapping up lecture exam 2, we're going to take that on Wednesday. So this is the last time we will meet before Wednesday. We are also wrapping up the first half of the course worth of laboratory structures. So of course I am in the lab today, and so I'm more than happy to show you guys models just so that you can see them one more time and ask questions about them before you take the test, before you take the lab practical. Okay, and so the lab practical will be available on Blackboard starting at 2 o'clock today, and it is due by midnight tomorrow. Okay. All right, so before we get into lab stuff, I do want to wrap up with the lecture so that you have a full 48 hours to kind of come to terms with this information. I did post, oops, goodness. I did post lecture notes for you guys, and we're just going to walk through them at this point. Okay. And as always, this is going to be on YouTube, just in case you missed something. Okay, so these are the lecture notes for today. These are the different topics that we're going to talk about. So we just wrapped up muscle physiology. Now we are going to talk about bone physiology. So pretty much how do they grow? How do they change over time? Both in the good normal ways, right, adapting to our lifestyle, and also how they might start to break down given what you choose to do with your life. Okay, so this is what we're going to be going through. Remember that bone is just another type of connective tissue. Remember, connective tissues have mostly extracellular matrix. That is, there's lots of proteins. There is always ground substance, and there's a possibility of having other types of substances as well. And in the case of bone and cartilage, which we're going to talk about because it is very similar to bone, essentially, there are inorganic molecules as well that can actually strengthen that extracellular matrix, right? So bone, of course, is a living tissue. It is very vascular, and we have these cells that are constantly building them up and breaking them down. And just to give you guys a little preview here, I have a short animation for you guys. A short animation that we will watch. Let's see, I think you can see this at this point, hopefully. And so here we go. This is an overview of everything that we're going to be talking about.
Our bones consist of an outer shell, compact bone, and an interior of spongy bone. Compact bone is dense and strong and provides an attachment site for muscle. Spongy bone is lightweight, rich in blood vessels, highly porous, and contains bone marrow, where blood cells are formed. While bone resembles cartilage, the collagen fibers of bone are hardened by deposits of calcium phosphate. There are 3 types of bone cells: osteoblasts, bone-forming cells, osteocytes, mature bone cells, and osteoclasts, bone-dissolving cells. Bone-producing osteoblasts form a thin layer covering the outside of the bone. The osteoblasts secrete a hardened matrix of bone and gradually become entrapped within it. They then stop secreting matrix and become osteocytes. Osteocytes are nourished by nearby capillaries and are connected to one another by thin extensions that osteocytes send out to each other through narrow channels in the bone. Although unable to produce more bone, osteocytes secrete substances that control the continuous remodeling of bone. Each year, 5 to 10% of all the bone in your body is dissolved away and replaced. This allows your skeleton to subtly alter its shape in response to the demands placed upon it. For example, by increasing the thickness of bones that carry heavy loads or are subjected to extra stress. Bone remodeling allows bone to be replaced as it ages and becomes brittle. As people age, the remodeling process slows, and bones tend to become more fragile as a result. The continuous turnover of bone also allows the body to maintain constant levels of calcium in the blood. Calcium from bones is retained in the blood if blood calcium drops, but returned to bone if blood calcium levels are adequate or high. This process is regulated by 2 hormones: calcitonin, which causes the bones to retain calcium, and parathormone, which causes bones to release calcium into the blood. Bone remodeling is the result of the coordinated activity of osteoclasts and osteoblasts. Osteoclasts secrete acids and enzymes that dissolve the hard bone matrix. Working in small groups, osteoclasts tunnel into the bone, creating channels that are invaded by capillaries and osteoblasts. The osteoblasts fill the channel with concentric deposits of new bone matrix, leaving only a small opening for the capillary. As a result of this process, in cross-section, hard bone is made up of tightly packed units called Haversian systems, each consisting of concentric layers of bone with embedded osteocytes. The concentric deposits surround a central canal through which a capillary runs.
Okay. Let's go back to our lecture here. So again, as the video showed you, bone is continually being remodeled. It is being built up and it is being broken down. And so that, of course, allows us to not only remetal our bones based on what we choose to do with our lives, but also it helps us to maintain a really important balance of calcium within our blood, right? And of course, we just finished talking about muscles, and of course, before that, neurons. And so hopefully at this point, you guys can appreciate just how important calcium actually is for the function of our body, right? In order to exocytose neurotransmitters, we need just the right amount of calcium in our body that is circulating freely in our body in order to contract our muscles, right? Our muscles have to have a certain amount of calcium inside of them, right, in order to release that and therefore bind to troponin, remove tropomyosin, and ultimately form the cross bridges. Okay, so maintaining calcium levels between a particular or within a particular range is a really important part to the functioning of our bodies. Okay. Bones in general are classified into categories based on their shape. We will be learning all 206 bones of the body. And, you know, generally we can appreciate these different shapes. For the upcoming lecture exam as well as lab practical, the only ones that we're focusing on are the axial bones, of course, minus the skull. That's at the very end of the class. And so hopefully this serves as a nice introduction to what different shapes are possible. Okay, so the long bones, oops, sorry about that. The long bones here, let's see. You can see this better. So the long bones are, of course, longer than they are wide. We have lots and lots of those, and we're going to be spending most of our time today talking about how they work. We have lots of short bones, right? So these are kind of cube-shaped and ultimately have little indentations for the formation of joints or articulations. We have some flat bones, mostly in our axial skeleton. So our sternum, for example, is a flat bone. Of course, it is flatter than it is wide or tall. We have irregular bones, right, which just have no particular pattern whatsoever. Our vertebrae are a great example of irregular bones. And sesamoid bones, we really have a pair of them. These are your kneecaps, right? So they actually form not in the same way that all of your other bones form. They actually form within a tendon, right? So we are going to be talking for the most part about how flat bones form as well as long bones form. Okay. So all bones have some basic structures in common, whether they are long or short or flat. They all have a periosteum, right? So remember we talked about this model right here. This outer coating on the outside of the bone is the periosteum, right? Around the perimeter, peri of the ost, the bone. Okay, and so no matter what bone you're talking about, all of them have a periosteum. This is a membrane that we're going to talk about in a little more detail in a couple slides here, but essentially this fuses to the bone, and this is where your muscles or your tendons attached to your muscles is actually going to attach. And so it's really important that this periosteum stays attached to the bone so that our muscles don't come off, and also we have stem cells in this layer, and so anytime we need to break down bone or build it up, maybe we've had an injury or whatever, we need that periosteum. And so it has to stay securely fastened to the surface of the bone, and in order to do so, it uses what are called perforating fibers, right? So these are actually embedded within the bone, and of course, if you take a look at this model here, we can see the perforating fibers attaching the periosteum right here. Okay. Of course, the bone itself contains both compact bone as well as spongy bone. The compact bone is the hard outer shell. The spongy bone, also called trabecular bone, also called cancellous bone, right? Why name something once? That is the spongy bone inside. And so here we can see a flat bone of our skull. This is the compact bone, and the spongy bone is kind of sandwiched in between. Okay. So let's talk a little bit more about the periosteum here. The entire surface of the bone is covered by the periosteum except for where the joints are. Okay, so actually, you know, at the top of your tibia, or at the bottom of your femur, for example, our knee comes together, and so those bones don't rub against each other and kind of grind and cause a lot of pain, a lot of discomfort, we have cartilage that protects the ends of those bones. So everywhere else except for the joint surfaces are covered with periosteum. There are actually two different layers of tissue within this membrane. First of all, we have the fibrous layer that is on the outside. It is made up of dense, irregular connective tissue so that we can push and pull on it in every which way, and it doesn't become damaged. Right? So if you look down here in this image, we can see that there are two different layers. Here's the fibrous layer on the outside, and the inner, the deeper layer that's actually touching the surface of the bone is the cellular layer. And so we can see that these cells look a little bit like squamous cells, right? So nice and flat. These are in fact osteogenic cells. So osteo for bone, genic for can generate anything that will ultimately become a bone or like a bone. That is, these cells can become those bone building cells, osteoblasts. They can become bone breaking down cells, osteoclasts. They can ultimately become osteocytes, right, mature bone cells, and I'll spell all these out for you here soon. And they can also become cartilage cells, and we'll see that bone and cartilage is very closely related. And so, yes, we have stem cells, right? So just like in our stratum basal, we are continually able to make more of these cells, and they can differentiate. That is, they can express different genes to become either bone building or bone breaking down cells. Okay. The periosteum also delivers about a third of the bone's blood supply, right? So we can see lots of blood vessels, right? So again, going back to this model, we see these perforating canals and these blood vessels ultimately distributing blood to all of the osteons, and we're getting there today. But these are all coming from the periosteum here. Okay. Also, there's lots of nerve fibers, so essentially the bone is very much alive. In general, long bones are broken down into the ends of the long bones and the shaft of the long bones. Okay, so on either end, proximal end or distal end, we have a fatter region of the bone. Okay, this is where the bones are going to form joints, right? So for example, this is a femur, which we'll get into in a couple days actually. But this entire structure here is going to form a joint with your hip, right? With your hip bones. And so these ends are fatter so that essentially the weight and the pressure can be distributed over a larger surface, and therefore not so much pressure on the bones themselves. Okay. Also, within these structures, the epiphyses, we have spongy bone, right? So here, up here, we have taken a section through the epiphysis. We can see lots of spongy bone inside. And so spongy bone, of course, forms a lattice work, and within that lattice is stored our red bone marrow, right? And as we're going to see, or as you're going to see next semester, whether it's the summer or in the fall, red bone marrow is where we make all of the formed elements of our blood. So the red blood cells, the white blood cells, the platelets, all of these things which are so very important to our existence are made within our bones, right? So bones are a lot more important than just keeping us standing upright and allowing us to move. They maintain calcium homeostasis. They maintain our blood supply. Okay. Epiphyses also, remember that they form joints or articulations. This is the official word for a joint. Because of that, they are covered with cartilage. And so anytime you see this blue color here or this blue color, that is showing us cartilage. The tissue type is hyaline cartilage, right? One of 3 different types of cartilage that we learned about previously. The structure is called articular cartilage, right? So hyaline cartilage is found all over the body, right? Our costal cartilages, for example, are made of hyaline cartilage. That's the tissue type. Articular cartilage is the structure. It is helping to protect the ends of the long bone within an articulation. Okay, so that's a really important distinction to make. Okay, so those are the epiphyses. In between the 2 fatter ends of the long bones, there is what is commonly called the shaft of the long bone, but more specifically, it is the diaphysis of the long bone. During the development of our long bones, this structure ultimately hollows out and forms a cavity within it. Okay, so this cavity is called the medullary or medullary cavity. It's essentially where the bone or the yellow bone marrow is going to reside. So yellow bone marrow is different than red in that in adolescents or adults, this essentially just stores some extra fat, right? And so it's not the kind of fat that you would go to the gym and try to get rid of. This is just an extra energy supply just in case we're running low, right? So you always have this unless you are literally starving, okay? You guys might have actually seen this in real life. So if you've ever, gotten soup bones from the grocery store to cook up for your dog or to make, soup for yourself or whatever, or even like the little slice of bone within a ham, there's that like gooey stuff inside the ring of bone. That gooey stuff is yellow bone marrow. Okay. Lining this medullary cavity is another membrane that is very similar in structure to the periosteum, but this is called the endosteum. So endo meaning on the inside. Inside of what? The ost, the bone. Okay, so in general, when in doubt, if you can't remember a structure having to do with the bone, tell me ost, and that will probably be partially correct. Okay. All right, a couple more features of the long bone that are essentially remnants or kind of like scars residual from the developmental process. First of all, we have these epiphyseal lines, right? So we can see that within the compact bone, within the spongy bone, there are these lines that look like compact bone, and in fact they are. These used to be your growth plates. Okay, so when your bones were first growing, this is all spongy bone, there were stem cells in here, there was actually cartilage, and ultimately your growth plates close, but those, like the location of those growth plates never actually disappears. It stays with you throughout your adulthood as a line of compact bone. Okay, and we'll talk about that in a few slides here. Also, there is a nutrient foramen, right, or foramen. This word foramen means hole, and so this is a hole for nutrients to get into the bone. And so if we take a look over here, we can see that there is a blood vessel that passes through the periosteum and is actually going to go all the way inside the bone into the medullary cavity here. This blood vessel penetration into the bone actually happens within the first 8 to 10 weeks of your life in utero, right? So 8 to 10 weeks after fertilization, these blood vessels penetrate into the kind of preliminary bone. It's not a bone yet, it's more cartilage, but essentially that blood vessel stays there, and it's going to continue feeding your bones for the rest of your life, right? So the periosteum supplies about 1/3 of the blood to the bone. The nutrient foramen with its associated blood vessels supply the other 2/3. Okay, and so some histology of bone tissue. Right? At this point, we have seen bone actual slides, right? So in the histology lab, we looked at some images of bone tissue. That was probably some of the easiest tissue to identify just because of those nice little ring structures. Right, the compact bone, just like we have seen in our model here, consists of these cylinders, and the structural unit of the compact bone are these osteons, right? Ost for bone, cylinders of bony tissue are osteons. And so we can see that essentially each one of these osteons is like a little tree, like a tree trunk, even with those little rings inside called lamellae. And they're very densely packed. So essentially the compact bone is a very densely packed forest of these little osteon trees. And so this makes the compact bone incredibly strong. In fact, about as strong as steel can be. Very strong indeed. Therefore, the structure matches the function, right? So we have essentially a shell around the outside of our bones that needs to be really strong because this is the weight-bearing structure of our bone. In fact, the weight-bearing structures of our body, right? So it has to be as strong as physically possible, okay? Each osteon contains 4 to 20 lamellae. Remember, lamellae are these layers here, concentric layers. we can see if we kind of pull those layers out, like in this image over here to the right, that the collagen fibers and the other things that we're about to talk about in these layers are arranged in a somewhat parallel fashion, but each layer has parallels or has fibers going in the opposite direction, right? So here we can see fibers coming up here. In the next lamella, we can see fibers going in this direction. And so this isn't by accident. This actually makes the osteons even stronger yet. So not only are these, or is this compact bone resistant to kind of like crushing or stress factors here, also we get a certain degree of twisting in our bones, particularly if you're younger, but a certain degree of twisting is possible because these fiber arrangements are strong to withstand that twisting motion. Okay. Also, all the way on the inside of these osteons, as we know from the lab, there is a central canal, and this central canal is where blood is delivered, used blood or deoxygenated blood is drained away, and there are also nerves as well, right? So actually controlling what's happening within the bone. Okay, just like that little video clip showed you, each osteon houses many what are called osteocytes. Now, when we learned about connective tissue for the first time, I told you that the suffix of the cell names would tell you kind of what it does, and that is absolutely the case. So the root site just means cell or mature cells of what? Of the osteo, of the bone. And so the osteocytes live within these little lacunae, right? So lives within these little cavities here throughout, you know, the entire life of the cell. And so again, kind of double duty here. We took a look at this model here before, right? So here's the osteons, here's the central canal. Each of these are lamellae. These little holes here are lacunae, and they house within them osteocytes. And so essentially, where do these come from? Well, these osteocytes used to be osteoblasts. Okay, so they used to be bone building cells, right? Again, just like the video showed you, that were so good at what they do, right? So good at secreting this bony matrix that they actually secrete it in front, behind, and all around themselves. So they actually get trapped in the very matrix that they themselves are secreting, right? So once they can't move anymore because they just buried themselves in bone, then they become osteocytes. Okay, so again, bone building cells are osteoblasts, right? The blast here indicates building, right? So building up, making more bony tissue. They will become osteocytes, right? The osteocytes live within lacunae. Remember that these lacunae, or these little cavities here, are connected by what are called canaliculi, right? So little tiny canals. And so essentially, in order to get nutrients and oxygen, right, from down here, for example, from this blood vessel, glucose, for example, has to be taken into this cell, pass through the cell to the next cell, pass through the cell to the next cell, and ultimately these canaliculi allow these cells to communicate and pass substances back and forth. Okay, so this is just another way of showing you this, you know, these structures. Remember, here are the perforating canals attaching the periosteum, which consists of dense irregular connective tissue as well as stem cells. Those stem cells can be osteoclasts, osteoblasts, osteocytes. Osteoblasts build up bone, they get trapped in that bone and ultimately become osteocytes. Those osteocytes live in lacunae, little cavities, which are connected by canaliculi. Right? So lots and lots of terms, and I repeat them multiple times today, not to be redundant, although I know it is, but most of these terms are also on your practical exam. And so I threw them at you guys last week and just said, you know, kind of learn these for now, but now you have a little bit more of the physiological background on these structures. So hopefully you can start to visualize them and recognize them a lot more easily. Okay. if we look closely at the compact bone, we can see that there are not only these concentric lamellae of the osteons, but also there are lamellae that go around the entire periphery of the bone, right? So these are called circumferential lamellae, which sounds so complicated, but they literally go around the circumference, right? So when you calculate the circumference of a circle, you're calculating, essentially the perimeter of it, right? And so the circumferential lamellae just add girth, right? They add diameter to the long bone. Okay, and so this adds a lot of strength, and it is formed by something called appendicular growth or appendicular bone deposition, right? So that's one of the things that we talked about today. I also, another structure that we talked about in the lab, right, on the models, are these perforating canals, right? So here are the blood vessels, and in order to get blood all the way in here, to the spongy bone, for example, we need to have these tunnels bored through, right, transversely through the osteons. Okay, and so that's compact bone. Let's talk about spongy bone now. So spongy bone is not actually spongy, right? It's not squishy like a sponge would be, but of course it looks like a sponge, right? If we look at this kind of honeycomb lattice type structure right here, it looks super spongy. And so spongy bone is always protected by the compact bone, right? The compact bone is the really rigid, really strong part of our bones because of the densely packed osteon arrangement. Spongy bone is not quite as strong, but it's not weak either. And so if you think about, back when we were on campus and you went into Big Blue and you look up and you can kind of see those rafters, or even just in the main academic spine, or even if you haven't ever been to Stockton, if you've been to any kind of like gymnasium, like high school gymnasium, if you look up, you'll see that there are these like this lattice type structure on the roof. These are called trusses, and essentially they are designed in such a way that they distribute the weight along these angles here, and therefore they can be super strong, but they don't have to be like a solid block of iron, right? And so this is great news, right? We can save resources, right? We don't have to either manufacture more of these thick iron beams or manufacture more trabecular bone, for example. But we still get the same benefits of the strength of a solid piece, a solid structure. Okay, and so in the same way that roofs can sustain a heavy snow load with this kind of structure here, that is exactly how our spongy bones work. Okay, so these this lattice work here, the trabeculae themselves, are distributing weight in a very organized fashion. It looks super disorganized, but it is nearly just as strong as the compact bone because of the particular arrangement of these trabeculae. Okay. Also, it is more organized than you would think, just looking at it. Over time, our trabeculae actually kind of arrange themselves, right? It's continually remodeling. So it arranges themselves to distribute the weight, to align along the lines of stress, right? So that's what this image is showing you up here. This again is a section through a femur. The weight of the body via the hip here is going to be borne by the femur, your thigh bone. And so in your knee, you don't want the entire weight of your upper body as well as your thighs coming down on like some little tiny space. So the region of the joint, the epiphysis, is super fat, right, in comparison, and the trabeculae within that epiphysis distribute the weight relatively evenly throughout your knee joint. Right? If anyone's had knee issues or know people with knee issues, which I'm sure is everybody, you know just how crummy it is to have knee problems, and so our body really tries to prevent that as much as physically possible. Okay. So again, the tissue itself, the type of bone is spongy bone or trabecular bone or cancellous bone. So that's the type of bone versus compact bone. the structures themselves, so the structural units of the spongy bone are the trabeculae, right? As opposed to the structural unit of the compact bone, which is the osteon. Okay, so that's really important, and that kind of sounds like a test question, right? Hint, hint, nudge, nudge. So the structure is the trabecula. The type of bone is the spongy bone. Okay, these are not arranged into osteons, but you do actually get these lamellae, right? You also have osteocytes within their lacunae as well, so really similar in that way. Okay, remember that that housed within the spongy bone is your bone marrow, right? And so you actually don't need really big, clunky, you don't really need blood vessels through a central canal, because this tissue is essentially surrounded on all sides by bone. Okay, and so just to remind you, all bones have these basic features in common. They all have compact bone on the outside and spongy bone on the inside, right? Whether you're talking about a flat bone or a long bone like this. And I do want to point out that in pretty much all of your spongy bone, you have bone marrow, right? But if you need to have your bone marrow sampled, right? So if you need to have it tested, you know, physician suspects that you might have leukemia, this bone marrow sample is taken usually from your iliac crest, so from your hip bone, like we can see down here, even though we have bone marrow in, you know, pretty much all of our bones, right? And so this makes sense, right? It's not just some random decision, random bone to take it from. Of course, if you try to take it from a flat bone, right, your flat bones are super skinny, and of course, if you miss and go right through, there's a lot of really important stuff that you might hurt around those flat bones. Right, your long bones are really strong. They have a really thick layer of compact bone, so that can be a lot more difficult as well. And of course, mobility of leg muscles and whatnot. And so kind of the easiest places to get bone marrow are your hips, right, which of course are right at the surface in the back there, as well as your sternum. Your sternum is an option. However, just like with the skull, if you miss on either side, there's a lot of really important stuff in your chest that you don't want to poke, right? So this is why generally bone marrow is taken from your hips, right? Just so you know. Okay, so that's the general structure of your bones. Let's zoom in a little bit further. What are all of these lamellae made of? What is compact bone and spongy bone actually consisting of at the tissue layer, at the tissue level? Bone is also called osseous tissue. We've been talking about ost. OSS is another root that means bone. Essentially, the small population of cells, right, all called osteo something, are surrounded by a ton of extracellular matrix, just like any other connective tissue. However, this type of extracellular matrix includes an inorganic matrix and inorganic matrix. So about 2/3 of the weight of your bones is actually made of, as you might imagine, calcium and phosphorus. So these salts, they actually spontaneously form little crystals around the existing proteins. These crystals are called hydroxyapatite crystals. They can be super strong in a direction, but also pretty brittle, right? There's no flexing whatsoever. Really strong, but can shatter when twisted, for example. And so the organic matrix actually allows our bones to be a little bit flexible as well as strong. And so the organic matrix, about 1/3 of your bone weight is made up of something called osteoid, right? Ost for bone. This is a lot of collagen, just like the dermis of your skin, for example, and a couple other types of proteins that really give rise to strength as well as flexibility. And so here, these are two fibulas, so part of your calf. If you treat the bone in such a way that you can get rid of the organic matrix, you have a strong bone that's super brittle. If you remove the inorganic matrix, you essentially have skin, right? So you have lots of collagen, can be pretty flexible, can give in a lot of different ways, but of course, this isn't going to keep your body upright. Okay, so it is the combination of these two, about 2/3 inorganic, 1/3-ish organic, that comes together to form your bone tissue. Okay, so we've used these words a little bit, but let's actually kind of walk through it in more detail at this point. The osteoblasts are the bone building cells. When the body needs bone to be built, and we'll talk about different scenarios where this would be the case, but when we need bone building, osteogenic cells, that of course are found within the inner layer of the periosteum, as well as the inner layer of the endosteum, right, coming from both directions here, they are signaled, right? So a ligand is going to bind to receptors on these osteogenic cells. That is going to trigger a pre-programmed response. Genes are turned on. They make new osteoblast cells. Okay, and so osteoblast cells, let's talk about that. Build bone by secreting some stuff, right? And specifically, they're going to secrete osteoid, right? So essentially the organic component of bone is literally made by osteoblasts, right? So we can see in this image here, borrowed from a peer-reviewed article, these are osteoblasts. They are going to transcribe, translate, and ultimately process these collagen proteins. Then they are going to secrete the collagen as well as all the other types of proteins in osteoid out of the cells, right? So some exocytosis going on here. So secretion, and these proteins essentially are going to weave together and arrange themselves in the optimal direction, right? Based on the needs of the body at the time. And at the same time, these proteins are organizing themselves into the appropriate direction. There are the osteoblasts are also going to be secreting enzymes. Right? Again, transcription, translation, processing through the RER, the Golgi, secretion via exocytosis of enzymes into the extracellular environment. And so these enzymes are actually going to be modifying the extracellular environment in such a way that it essentially like calls over more salts. Right? So salts being calcium and phosphorus that spontaneously crystallize around these existing protein fibers. Okay, so again, osteoblasts secrete collagen and other types of structural proteins called osteoid. They also secrete enzymes which call more salts to the scene, which then spontaneously ossify around the osteoid. Right, so now we have 1/3 osteoid, 2/3 hydroxyapatite crystals. As we know, these osteoblasts are just so good at what they do that they are, secreting their osteoid, which is then ossified. And this isn't just happening like in front of or behind them, it's also happening in front of them as well. And so they ultimately bury themselves in this bony matrix. Okay? And of course, once they're trapped there, they become osteocytes. And so they're no longer building bone, they're no longer secreting osteoid. Instead, they are merely maintaining it. Okay. And finally, the third type of bone cell are the osteoclasts, right? So bone breakdown seems really scary. It seems like a really bad thing, but we absolutely need bone breakdown. Okay? We need to continually remodel our bones and, of course, take calcium out of reserves in our bones and dump it into the blood if necessary. And so if we need to break down some bone, again, we'll talk about those conditions later, but if we need to break down bone, essentially those osteogenic cells, those stem cells, turn on particular genes which make them osteoclasts. What do osteoclasts do, or what do they look like? Well, essentially they are these big multi-nucleate globs with, and so anytime you see a lot of nuclei, this essentially means that there is an even greater capacity for transcription and translation. Therefore, lots and lots and lots of proteins can be made. And so if we zoom in here to the edge of this osteoclast, we can see that there are tons of enzymes that are being secreted onto the surface of the bone. These enzymes, as well as acid, right, so extra hydrogen ions, is actually going to eat away, it's going to break down that bone, therefore mobilizing things like calcium and phosphate back into the blood. Okay, so again, in this image here, we can see the periosteum. This cell is just differentiated into an osteoclast, and now it is going to be spitting acid and enzymes onto the surface of the bone and essentially burrowing farther and farther and farther into that bone. Okay. Of course, these mobilized substances can be released into the blood. So if your blood calcium levels drop below a particular threshold, we need to start breaking down our bone. Okay. And so there's always some degree of bone building by osteoblasts and bone breaking down by osteoclasts. And as long as we maintain this balance, we will have both strong bones as well as enough calcium in our blood. It is only when you start to shift this balance between osteoblasts and osteoclasts that you can start to have some trouble. Okay, so let's talk about where bones come from to begin with, right? And it all ties back to those three types of cells. Those are the main players in these stories. The general process of making bone can be called either ossification, right, which is literally the forming of crystals on osteoid, or more generically, we can say osteogenesis, right? We are generating osteo, or we're generating bones. This process starts when you're an embryo, actually, so very early on in development, and most of your bones exist by the time you're 7 years old, so 7 years post-birth, of course, but they don't stop growing, they don't stop changing. Okay, so again, this initial development of your bones starts very early on in your development, and then it's going to continue throughout your life. The very first thing that happens is pretty much just like quick and dirty, we need some kind of model for our bones, right? It doesn't have to be perfect, it doesn't even have to be strong yet, right? So a fetus isn't actually experiencing any pressure, right? It's floating in amniotic fluid, and therefore the bones don't need to be strong. We just need to have some kind of bones. And so the very first thing that happens is we form this really haphazardly almost thrown together primary or immature or woven bone. So essentially like spongy bone, but without the kind of the thought put into it, right? There are two different processes that start with this and then proceed from there to actually reorganize the bone and increase its strength. First of all, we have intramembranous ossification. So ossification, right, so calcifying your osteoid, and intramembranous, so this is happening inside a membrane. What membrane? Remember that all connective tissue is derived from mesenchyme connective tissue. That's a way back to exam 1 material. In intramembranous ossification, we build our flat bones and our sesamoid bones from directly from a connective tissue sheet, right? And in this image of an embryo here, we can see some of the skull bones forming. Literally, there was a membrane around your soon-to-be brain, and bones literally form within that membrane. The other bones, right, so your long bones, for example, form via something called endochondral ossification. Right? So again, this process of crystallizing calcium and phosphorus, but this time, instead of forming directly from a membrane, they form from a cartilaginous model. Right? So endo, inside, chondro, remember, means cartilage. So inside the cartilage. In this image down here, we can see very early bones. We can also see our tail, right? That's a thing, we have that. So these here aren't bones yet. They're just cartilaginous models of a bone, right? And so in both of these, intramembranous and endochondral, we start with just irregularly arranged collagen bundles, osteocytes, osteoblasts, building bone as much as they possibly can, as quickly as they can, just to get the models there. And this process is going to continue to remodel them over time. Okay, usually that really disorganized model is almost immediately broken down by osteoclasts and arranged much more strongly by osteoblasts. Okay. And so intramembranous and endochondral ossification are both forming bones out of existing mesenchyme or cartilaginous tissues. Appositional growth is increasing the girth, it's increasing the diameter of the bones that already exist. So appositional can only happen to an existing bone, not to membrane, not to cartilage, only to existing bone. Okay, so let's walk through briefly the steps of intramembranous ossification. All right, so here we see a fetal skull. Okay, if we zoom into that fetal skull, we can see mesenchyme tissue. Right, so here are stem cells, right, that can ultimately differentiate into any of the connective tissue cells. And we can also see lots of like collagen fibers and whatnot around here. So, generic old connective tissue. Given a particular signal, which we're not going to get into, but ligand binds the receptor, these stem cells then differentiate into osteogenic cells, right? And so this is actually turning on particular genes that essentially say, I'm going to become something that has to do with bone or cartilage and nothing else, right? So I'm not going to become a blood cell, I'm not going to become an adipocyte or anything, only bone or cartilage cells from here on out, right? So then these osteogenic cells would further differentiate into osteoblasts, okay? And so this is actually happening in a couple different places throughout this membrane that is covering your soon-to-be skull. Okay, and so we can call these little areas of osteoblast differentiation, we can call them ossification sites or ossification centers. Okay, and so osteoblasts are going to start doing what osteoblasts do. They're going to start secreting osteoid, which is that organic matrix. They're going to secrete those enzymes, and essentially spontaneous ossification is going to form, of course, trapping some of those osteoblasts in the process, right? So what we can see here is one of these ossification centers. We started off with just normal old stem cells, then we made osteogenic cells, then we made osteoblasts, right? And now we're starting to form this bony tissue, immediately trapping the osteocytes inside, okay? Okay, we can also see that on all sides of this little ossification center, there are more osteoblasts, right? So these osteoblasts are continuing to differentiate, and they're kind of moving out in all directions, right? Just like that video showed you, that they kind of like tunnel through. This is therefore building bone out in all directions. So if we look at this parietal bone right here, an ossification center might happen or occur within the very middle, and then osteoblasts are going to start moving out, building bone in all directions, and therefore the bone gets bigger and bigger and bigger. Okay, intramembranous ossification as a process begins at about 10 weeks post-fertilization, right? And that's important because between 8 and 10 weeks post-fertilization, your cardiovascular system really starts to develop and differentiate. And so after 10 weeks, you have blood vessels. And so connective tissue is generally very vascular. And so as these osteoblasts are building their bone outwards in all directions from their ossification centers, they actually have to weave around the existing blood vessels. And so this naturally forms something that looks a heck of a lot like spongy bone. So it's naturally forming trabeculae, right? So that's what these images over here are showing you. weaving around the blood vessels, forming a lattice, a trabeculae. Okay. Now, while these trabeculae are forming, we essentially differentiate some more of those mesenchymal stem cells into what is going to become the periosteum. Right, so over here, we can see that these blood vessels are organizing and that these extra cells here have differentiated into periosteum cells. And so they are going to actually start forming bone, right? So more osteoblasts, forming compact bone, sandwiching on either side of the spongy bone. Okay, so again, we form a periosteum. The deeper layer of the periosteum contains stem cells. Those stem cells become osteoblasts and start secreting osteoid into this nice compact bone structure here. Right, so this entire process is happening in all of your flat bones simultaneously, and so your flat bones like continue to grow towards one another, right? So out from the ossification center, but towards the other bones. And so this here hopefully will help you to visualize that. Here is a parietal bone. Here is one half of the frontal bone. This might be an ossification center. This might be an ossification center. And so essentially this bone building by osteoblasts, as well as the simultaneous formation of the periosteum, is going to happen out in all directions. And so this bone is growing towards the front. This bone is growing towards the back, right? So they are, you know, the bones themselves are getting bigger and they're also getting closer together. Okay, this process absolutely does not stop when you're born, and I'm sure that you guys can all appreciate this. Babies have soft spots. These soft spots are actually called fontanelles, and what the soft spot is the mesenchyme membrane that is still kind of being converted into or having bone formed inside of it. Intramembranous ossification is still occurring within this mesenchyme tissue. And so this, of course, is really important because baby's head is not the size of an adult head, and so you still need you know, your bones to be able to grow to accommodate your growing brain. Also, the particular position of these joints, right, so where this membrane is as opposed to bone, is really important in the birthing process. So essentially the fact that there is a joint that isn't filled in, right, so it's still membrane as opposed to bone, means that those two halves of your skull when you're being born can actually pass over one another to make baby's head a little bit narrower to ease its passage through the vaginal canal. And then after birth, right, you can watch it just pops right back out, right? So kind of a cool side effect of incomplete intramembranous ossification. Okay, so that is probably the simplest process here. The next process I want to talk about is endochondral ossification. This is happening in pretty much the rest of your bones. This process starts at about 8 weeks post-fertilization. So in fact, before intramembranous ossification is going to begin. And this also differs in that this process has to start from an existing cartilaginous model, right? So in utero, you form cartilage, vague shapes that look kind of sort of like bones or what's going to be bones, but it's just cartilage. Okay, and so endochondral ossification starts at 8 weeks post-fertilization and is going to continue through puberty and beyond. And pretty much those cartilaginous models are going to be replaced by bone with some exceptions. That is, we keep some hyaline cartilage, we don't turn it into bone in our chest, right? So this allows us to flex our rib cage and therefore inhale and exhale a lot more easily. Also, the ends of the long bones maintain that cartilage, right? So just like I mentioned before, that cartilage is made of hyaline cartilage tissue and it is called articular cartilage. Okay, so we have looked at cartilage under the microscope before. But really, what is this type of tissue? So cartilage is a gel of proteoglycans, so essentially different proteins, different carbohydrates, and they form this like brush-like structure here. And this is important because these brushy structures trap water molecules and essentially creates a nice cushion, flexible cushion that is still really strong surrounding the cartilaginous cells. And so this structure is highly resilient, right? So again, it covers our long bones, right? You do jumping jacks and your femur would grind against your tibia if not for this cartilage. Unlike most connective tissue, cartilage is avascular. That is, it doesn't have blood vessels or many blood vessels, if any in some places, and it has very little nervous control as well. And so what that means is that in order to get nutrients, oxygen, etc., to, for example, this cell, these substances have to diffuse from this cell through the matrix to this cell, through the matrix to that cell, through the matrix to that cell, right? So this is a very time-consuming process. And so these chondrocytes, these cartilage cells, can't really have a big energy demand. If you damage your cartilage, they take a really long time to get the materials they need to actually repair that cartilage. And so if you wreck your cartilage, if you grind that cartilage away, it will take a really long time to heal, or it won't heal at all. Okay. Again, cartilage and bone are pretty similar in a lot of ways. One of those ways is that there is a membrane surrounding the entire structure. However, the perichondrium, right, so peri for perimeter, chondro for cartilage, this perichondrium might appear to be like the periosteum, but it also has a function of essentially keeping that gooey gelatinous matrix in shape, right? The bone has its own shape, right? If you happen to rip off the periosteum, it's still going to look like a bone. Perichondrium essentially is acting like a girdle, right? So when femur comes down on tibia, when you're doing jumping jacks, the reason why that cartilage doesn't ooze out to the side is because of this girdle, because the perichondrium is keeping that cartilage in the correct shape. Okay. Also, the perichondrium, just like the periosteum, contains blood vessels. So this is the only source of blood that the cartilage is going to get. Okay. And again, just like the bone, we have these specialized cells that live within cavities that are essentially sustaining the surrounding tissue. And so it's not in bone, so we can't call it osteo whatever. Instead, we have to call it chondro whatever, right? So chondro for cartilage. Chondroblasts build cartilage, just like osteoblasts. They get stuck in that cartilage, and then we call them chondrocytes, right? Mature cartilaginous cells. And again, just like we can see here, there are little cavities called lacunae. Okay, so lots in common. Again, we looked at these tissues for the last lecture exam. These things are still applicable to your practical exam, which opens up this afternoon. And also, we're expanding on this a little bit because it's so very relevant to this chapter. 3 types of cartilage. Most common is hyaline cartilage. We also have fibrocartilage, and what's different about fibrocartilage is that we can see not only are there these proteoglycans, those nice like brushy protein carbohydrate conglomerations, but also we have a lot of collagen, right? So these big dark lines here are collagen fibers. So this is super strong and particularly resistant to damage. And so fibrocartilage is found in places that get a lot of wear and tear, such as your intervertebral discs, right? And also the discs of the knee, so the menisci or the meniscuses of your knee, which we'll look at beginning of next week. Okay. And finally, elastic cartilage, same deal as the other 2, but instead of just plain proteoglycans, instead of proteoglycans with collagen, now we have proteoglycans with elastic cartilages. with elastic fibers, that is. And so your external ears, right, are nice and flexible and really strong and whatnot, because it's made of elastic cartilage. Okay, but you guys already know that from the last section. Right, and of course, hyaline cartilage is just pretty. Okay, so in endochondral ossification, we begin before weeks post-fertilization with cartilaginous models made of hyaline cartilage. Right? We are going to be walking through these steps over here, and of course, I number them for you here. Unfortunately, I've never found a great animation of this. If you find one, by all means, let me know, but we're just going to walk through the diagrams. Okay, so we start with this blue thing that looks kind of like a long bone in that it has fat epiphysis ends and a skinnier diaphysis shaft in the middle. Okay, it's also blue, which implies cartilage. And so the very first thing that happens about 8 weeks post-fertilization is that some of those chondrocytes that are trapped within their lacunae are going to grow. Right? So as they grow, they are going to be pushing that lacuna bigger and bigger and bigger. And so you can see down here, there are like tiny little dots. Those are normal old lacunae. When some chondrocytes start to get bigger, so too do their cavities. And so bigger and bigger and bigger cavities, and then the chondrocytes die. Right? So essentially they disintegrate, leaving these big gaping holes in the very middle of this cartilaginous model. Okay? At the same time this is happening, we're getting more blood vessels, right? Remember I told you that between 8 and 10 weeks, we start to get more and more blood vessels, and that's important in our intramembranous ossification, but also during this period, we are growing blood vessels around the diaphysis of each and every one of these cartilaginous, soon-to-be bones. Okay? When the blood arrives, it brings with it not only oxygen, not only nutrients, but also a lot of chemical signals. And these ligands are going to trigger the differentiation of stem cells within this connective tissue. Okay, that is, ligand binds to receptor, triggers certain genes to turn on, and essentially what we're going to get here is stem cells that were in the perichondrium, right? So this girdle around the long bone, or soon to be long bone, they are going to turn on different genes to become osteoblasts, right? So as soon as we get cells that turn on the osteoblast genes, we can now call this membrane the periosteum, okay? And so just like osteoblasts are prone to do, they are going to start secreting osteoid, which is then ossified, specifically around the diaphysis. So what we have now is a cartilaginous bone-shaped structure. In the diaphysis, you have lots of big gaping holes where chondrocytes died, and also you have this shell of bony tissue surrounded by periosteum just around the diaphysis, right? Remember, this all happened because the blood vessels brought chemical signals that said, hey, this needs to be a bone, not cartilage. Okay? And so we continue from here. The blood, of course, doesn't stay on the outside, right? As we talked about earlier today, the nutrient foramen is what allows blood vessels to enter into the diaphysis of the long bone. That happens now. Okay, so here we have blood vessels actually penetrating through that nice shell of bone around the diaphysis. Okay, and of course, with the blood comes ligands, and those ligands are going to trigger stem cells inside the diaphysis to differentiate into osteoblasts. And of course, the osteoblasts are then going to start secreting osteoid, which is then ossified. Remember that there are all these big old holes in the diaphysis. As a result, there's space for bone tissue to actually grow. Right, so we start filling in those holes, sending out osteoblasts in all directions. They're just sloppily throwing down any osteoid in any direction. We want to convert this into bone as quickly as possible. We'll go back and reorganize it later. And so this process is going to continue, right, in all directions, right? So out towards the edges, right, towards that shell of bone, and up towards the epiphysis and down towards the epiphysis. Okay, while this kind of primary or immature bone is being laid down as quickly as possible, we simultaneously get the differentiation of osteoclasts and osteoblasts again in the diaphysis, right? So what's happening here is that osteoclasts are going to break down that sloppy bone and allow new osteoblasts to more slowly reorganize that diaphysis. And so this is going to lead to the formation of the medullary cavity, right? So the medullary cavity in adults is filled with yellow bone marrow. As a fetus and as a young child, this is actually filled with even more red bone marrow, because of course growing takes lots and lots of energy, lots and lots of blood, and so we need as much bone marrow as we possibly can. Okay. Also, kids have to eat a lot all the time. They don't have a lot of extra energy stored in these places yet, and so they need to take it in continuously. Okay. All right, so while this reorganization is occurring within the diaphysis, right, so the formation of the medullary cavity, now filled with red bone marrow, soon to be filled with yellow bone marrow, we are going to get the formation of the secondary ossification centers. Right? So the primary ossification center was the entrance of this blood vessel into the diaphysis, formation of bone here. Now we are going to get more blood vessels, and couple into each epiphysis, that again bring these osteoblasts. Osteoblasts are going to start secreting osteoid, which is then ossified, and of course this is happening in all directions, spreading out towards the edges of the epiphysis. Okay, and so bone growth, bone formation is still happening here, and now it's happening in the ends of the long bones as well. Okay, and so soon these epiphyses are going to fill with spongy bone. Right? We can reorganize that as we go along. And we're still getting bone formation within the diaphysis. And so if we look at this next image over here, we can see that there is actually a separation between bone formation here and bone formation up here. And so there is purposely maintained a thin layer of hyaline cartilage between these two bony structures. This cartilage line, this cartilage plate, is called the epiphyseal cartilage, or the epiphyseal plate, or you've surely heard it referred to as the growth plate. Okay, so remember that endochondral ossification has to start with cartilage before we can form bone. So even when the entire initial little tiny fetal bone is replaced by, or fetal cartilage is replaced by bone, we still need to get bigger bones and these short little things. So growth is going to continue from the hyaline cartilage that is maintained between the epiphyseal bone and the bone in the diaphysis. Okay. And so let's zoom in on this even more. Keep in mind that we are just looking at, for example, the proximal epiphysis, but this is also happening in the distal. That is, bone growth is occurring up, making you taller by going up, but also making you taller by growing down as well. So it's happening on both ends of the long bone simultaneously. Okay. How do growth plates work? Well, on the epiphyseal side, we have the growth of hyaline cartilage. Right? So essentially we have chondrocytes that are dividing, right, and going mitosis, making more of themselves. These are going to build more cartilage up towards the epiphysis, or down towards the epiphysis, right? So towards the epiphysis, right? So we're building cartilage on this side right here. Just like we saw in the very beginning, existing chondrocytes, which are trapped within their little lacunae that they themselves did, they're going to die. Okay, and so they're going to die. They are going to decompose, leaving these big open holes. Now, these holes are very convenient for the osteoblasts. Now, keep in mind that the osteoblasts are chugging on up the diaphysis towards the epiphysis, laying down osteoid. It is then ossified. They are trapped within this bony matrix. Okay, and so they are proceeding towards the epiphysis. They encounter this big gaping hole, which used to contain a chondrocyte. They are going to fill it with bone, and they themselves become trapped. Okay, we make more osteoblasts, and they continue working their way up. And so essentially, the chondrocytes are going to be building more and more and more cartilage up towards the epiphysis, while the osteoblasts are coming behind them, filling all of that cartilage in with bone. And so for a good portion of your childhood, both of these are growing at approximately the same rate, right? So we build cartilage, we replace it with bone. We build cartilage, we replace it with bone. Okay? And so this is just another way of showing you this. If you were to look at some histology, this is what you would see, right? So the proliferation zone, we make more chondrocytes, which make more cartilage. Those cartilaginous cells die, leaving big holes, and those holes will then be filled in by osteoblasts. Okay, and so ultimately, this is going to allow our bones to grow in both directions. So, you know, on Monday, your bone is this length, and because the cartilage has built on this side, the bone is built on this side, essentially, Monday the next week, your bone is this tall, right? So again, this is going to continue. You know, the speed of the chondrocytes is approximately the same speed as the osteoblasts, and both are going to be growing in both directions. As we're all surely familiar, there are certain periods where growth spurts are pretty darn common, particularly when you hit puberty, right? So, you know, you might go home in June at the end of a school year in the 7th grade and come back in the 8th grade the next year, and all of a sudden you're 6 inches taller. Oh my gosh, what the heck just happened? Hormones are going to ramp up this process quite considerably. And so, as I said, normally chondrocytes and osteoblasts move up at approximately the same rate. Sex steroids, so the hormones that are associated with the process of puberty, are going to ramp up particularly the activity of osteoblasts. And so the chondrocytes are still going to be doing their thing at approximately the same rate, but now the osteoblasts are going to hurry up, and they're actually going to meet those chondrocytes before they even die, right? And so what this means is that there's no more cartilage being laid down, okay? Without cartilage, we cannot complete endochondral ossification. So when your osteoblasts catch up to your chondrocytes, this is when your growth plates close, right? So this is when there is no more cartilage to convert, and therefore we're left with this little epiphyseal line, this scar that used to be the cartilaginous epiphyseal plate, and now it's not. Okay? And so, of course, if your osteoblasts are just charging through and making bones super fast, that is how you can get several inches of growth in a relatively short period of time. Okay. I also want to point out this X-ray right here. You can see the growth plates open. Keep in mind that this is cartilage. Cartilage doesn't show up on an X-ray, and so it looks like the epiphysis is actually separated from the diaphysis. Okay. So that's you can tell when your growth plates are open or closed. Okay. And of course, the other thing I want to point out here is that this isn't just happening in your femur, right? It's not just happening in your legs or your arms. This is happening in every long bone of your body. And so each and every little phalanx here has its own growth plates. Okay. And so, as I said, puberty really ramps this process up. It makes you grow super fast. But different bones, epiphyseal plates are going to close at different times. And so this just gives you some very basic ranges. Now that you'll have to spit these numbers out at me, I just want you to kind of keep in mind that maybe some of your growth plates aren't even closed at this point. So, you know, maybe your ankles will close when you're a teenager, right? Within a few years of puberty, your osteoblasts finally catch up to your chondrocytes. But then other bones, like your clavicle here, might not close until you're like 30, right? And so, at the beginning of this class, I had a little image that said, you know, pretty much you are what you eat. Literally what you put into your body is going to affect your bones, right? What you're doing at this point is affecting how much your bones are growing, how much, you know, what's going into them. Right? So just a little side note there. Okay, so intramembranous ossification forms your flat bones and sesamoid bones from a membrane, from a mesenchyme membrane. Endochondral ossification forms your long bones from cartilaginous models. We need cartilage in order to make bone in endochondral ossification. There is a third type of bone growth, or bone deposition in this case, that occurs but only from existing bones. And so this is happening throughout your life, right? Whether you are, your growth plates are closed or not, this is always happening. And so appositional bone growth allows your bones to grow in diameter and not just in length, right? So if you can imagine a fetal femur, for example, is probably like this wide, but that is certainly not wide enough to sustain the weight of an adult body. And so we not only need to grow in length, we need to grow in width. And so what we can see down here is appositional bone growth. Here's an infant bone. What we do is we have of course removed bone with osteoclasts on the inside. This widens your medullary cavity. At the same time, we have deposition, bone growth by osteoblasts around the perimeter. And so remember that the periosteum contains stem cells. They're there throughout your entire life. And so if your body is putting more weight on your bones than your bones can withstand, essentially osteogenic cells will differentiate into osteoblasts, and they will deposit osteoid in a ring around the outside of your bone. Okay. Now, of course, your bone is wider now, and so we can clear out a little bit more space on the inside with osteoclasts. And so this process happens over and over and over again until we end up with an adult bone, big, wide, yellow marrow-filled medullary cavity with lots of these extra rings or lamellae around the perimeter. And so we've actually already seen evidence of appositional bone growth before. All right, so going back to this model here, we have the osteons, right, and the osteons have all these lamellae, but also around the outside, right, around the perimeter, we have multiple layers of bony tissue, and these are called circumferential lamellae. Right, so they're going around the circumference, and again, they are made by appositional bone growth. Right, and the other thing this image shows you is the fibrous layer of the periosteum and the osteogenic layer of the periosteum, which is where the osteoblasts come from. And so that's why when they differentiate, they can lay down bone right here. Okay. Once again, your bones, they'll just grow and then stop changing. As the video showed, your bones are continually remodeling themselves and oftentimes continually repairing themselves. Again, there's usually a balance of bone building and bone breaking down, or bone deposition and bone resorption. Okay. We can change this balance if we have mineral needs. That is, if we need more or less calcium, if we need more or less phosphate, we can actually dip into the reserves in our bones to help the body maintain homeostasis. If there's damage, right, so if you break a bone, you need to fix it, right? So obviously you don't want to have equal amounts of bone breaking down and bone building. You want to have a lot more building to repair that gap. And of course, disease is an unfortunate imbalance of these two processes. Right, so let's talk about homeostasis first. This pie chart here shows you what is actually in your bone. It is a reserve for the overwhelming majority of your body's calcium as well as the body's phosphate. And as I talked about earlier today, calcium is so important for your neurons, for your muscles, for everything, that we need to have just the right amount of calcium. Okay, we have lots of calcium, right, within our bones so that we can always break down more bone to increase the levels in our blood. Okay, this is a classic homeostasis diagram. It shows you calcium homeostasis. So when the body is within its target range, right, we have a balance, right? So the target range is 9 to 11 milligrams per 100 mils of blood, right? So we're shooting for 10-ish, right? If we have a ton of calcium, right? So you just had a glass of milk, Your digestive system pulls calcium into your blood, and so now you have increased your blood calcium levels over 11. Okay, what do? That's also bad. Okay, so if there is an imbalance where there's too much calcium, our thyroid gland detects this. Okay, the thyroid gland, in response to high calcium, is going to release calcitonin, right? Because there's a ton of calcium. Calcitonin is a hormone that is detected on several different target tissues, including our bones. In response to calcitonin, bones are going to take more calcium out of the blood and build more bone. Okay, so ultimately, this should bring calcium back down to about 10. Okay. On the other hand, if you don't drink milk in a while, but of course you're still using calcium in your body, this is going to decrease the calcium levels in your blood below 9. And this is detected by your parathyroid glands, right, which are actually kind of on the posterior side of your thyroid, which is like right here in your throat. In response, parathyroid glands release PTH, or parathyroid hormone, or parathormone, as the video said. PTH is another hormone that is also detected by many target tissues, including your bones. If we don't have enough calcium in our blood, we can tell osteoclasts to start spitting out more acid and enzymes and break down that bone even faster. Okay, so let's dive into this just a little bit more. We'll start with low calcium levels. Again, if you don't have enough calcium in your diet, right, you forgot to take your vitamins this morning, whatever, your calcium levels might drop below this target threshold, right? There is a negative feedback process which involves the parathyroid gland releasing parathyroid hormone, or PTH, or parathormone. Right? Any of those would be okay for me. Of course, this hormone circulates throughout the entire body, but there are only receptors for PTH on certain organs, and these organs all have some kind of a role in maintaining calcium. First of all, and most relevant to us right now, is the bones, right? So if our bones bind to PTH, what that's going to do is trigger osteoclasts to start working more to break down the bone, right? So essentially immobilizing that calcium that has been ossified onto the osteoid, and that calcium is released into the blood, therefore increasing the calcium a little bit, right? But that's not our only response to low calcium levels. We can also tell the kidney that we have too low calcium, right? So kidney cells also have PTH receptors. In response to binding to PTH, the kidney is going to do two different things. First of all, it's going to reduce how much calcium we pee out, for example, right? So we actually are reabsorbing more calcium from our filtrate, or our soon-to-be urine, back into our body, and therefore back into the blood, right? So we stop losing so much calcium in the urine, and instead we put it in the blood, correct? Also, the kidneys secrete more of its own hormone, right? The kidney actually has tons of hormones that it detects and it produces. One of those is calcitriol. And calcitriol tries to increase calcium, right? It's actually a derivative of vitamin D. And so calcitriol, as well as parathyroid hormone, are both detected on the cells of the intestines. When these are detected, and actually calcitriol actively has a role in this process, it is going to increase how much calcium we absorb from our food, right? So if your calcium levels are super low, you want to make sure that every little ion that is in your food actually comes into your blood, okay? So again, all of these target tissues are working towards increasing the calcium in your blood by breaking down bone and immobilizing stored calcium, by taking more calcium from your food, and by releasing less calcium in your urine. Okay. All right. And so on that note, calcium, as I said, is a derivative of vitamin D. So you've all surely seen that whole milk, for example, now has vitamin D in it. Well, the cow is not putting vitamin D in the milk for you. That has actually been added as a supplement because you absolutely need vitamin D in order to absorb calcium from your diet, right? So even if you're drinking milk constantly, if you don't have vitamin D / calcitriol, you're not actually going to get that calcium into your body. And so there's a couple different places where you can get vitamin D, as I said, from your milk, for example, that has been added there somewhat recently. But of course, there's other ways of getting it as well. So from your food, you can get vitamin D, which is cholecalciferol. You can also get vitamin D from being in the sun, as you've probably heard of before. Essentially, the UV rays from the sun configure a steroid that is in your skin in a particular way that it actually forms automatically this particular substance. So cholecalciferol, that is vitamin D. This molecule is then processed by the liver to a certain extent, which is then sent to the kidney, and the kidney finally releases this product. Again, derivative of vitamin D. Without vitamin D, you don't get calcitriol. Without calcitriol, you cannot pull calcium out of your food and into your blood, right? So even if it's in your diet, you're not actually able to get it. And so why do we put vitamin D in the milk? Well, for a long time, calcium imbalances such as rickets were a fairly regular occurrence. So rickets is a condition where there's either not enough vitamin D, you're not getting enough vitamin D, you're not getting enough calcium, or some kind of a genetic issue where you can't actually use it. But essentially, you're not able to use vitamin D to get calcium, and without enough calcium, you cannot strengthen your bones. So essentially, you end up with more frequent fractures, right? Because we don't get this ossification as strongly. And also another characteristic rickets symptom are bowed femurs, right? Bowed legs in general. Remember a few slides ago, we took a look at what you could do with a bone with organic matrix versus without inorganic matrix. And so essentially, if your bones don't have enough calcium, they essentially become more like skin, right? So they're super flexible, and so they can't support the weight of your body, and so they actually grow bowed out to the side, unfortunately, with the lack of calcium. Okay. All right, so on the other hand, what happens if we have too much calcium? Yes, we can have that problem as well. So if there is high calcium, you just had lots of milk and it had lots of vitamin D in it, or you've been out in the sun a lot because it's lovely, you're gonna have lots more calcium in your blood. And so the organ responsible for detecting this is the thyroid gland. In response to high calcium, we release calcitonin, right? So when there is a ton of calcium, you release calcitonin, right? So lots of a ton, okay? Calcitonin, another hormone, is going to be detected on the same 3 target tissues and produce like the opposite effect as PTH. Calcitonin is going to stop the osteoclast from breaking down your bone, right? So we don't need to keep mobilizing more and more calcium. We've got plenty. Okay, and so we stop having bone breakdown. We might increase bone deposition a little bit, right? So start pulling more calcium out of the blood and putting it into the bones. Just in case you stop drinking lots of milk with vitamin D, you will still have a lot of calcium in reserve. Okay, calcitonin is also going to tell the kidney that there's a ton of calcium in the blood. And as a result, the kidney is going to pee out more calcium, right? We don't need it in our blood, and so we can put it into the urine. And it is going to release less calcitriol. Remember, calcitriol tries to increase calcium, and so with less calcitriol, we are absorbing less calcium from our diet, and more is released in the feces. Okay, so ultimately, more calcium exits in the feces and urine, more calcium is deposited in the bone, and the blood levels are going to decrease. Okay, another time when we want this balance between osteoclast and osteoblast activity to shift is when we break a bone. Right? So a fracture is a crack or a break in the bone. And so this process very much resembles endochondral ossification, and it requires the stem cells excuse me, within the periosteum and or end ostium. And I'm going to try to play this from here. It's just a short clip that super helpful. Just kidding. Every time I think it's going to work. Nope. So let me show you the other way. All right, guys, and we are almost done today. All right, so fractures. How do we heal them? How do we replace that bone?
When a broken bone or fracture occurs, the body leaps into action to begin the healing process. When the bone breaks, it bleeds, forming a mass of blood known as a hematoma. The hematoma is rich in cells that are capable of growing new cartilage and bone. These cells set to work forming a callus to hold the bone fragments together. The callus is at first more cartilage than bone. This is known as a soft callus. The soft callus gradually begins to fill with bone, evolving into a more stable hard callus. After the hard callus is formed, the bone may not look like it did before the fracture. It may look slightly crooked. It probably has a large bony callus. Over months and even years, the bone begins to remodel based on the stresses it is exposed to. Remodeling allows the bone to take on a shape similar to its original shape. If there is too much displacement or angulation, meaning that the bone fragments are pushed too far apart or the bone is bent too much, remodeling may not be sufficient. The bone may heal crooked or may not heal at all. This is why fractures sometimes need to be reduced or set. Once the fracture is well aligned, it is important to keep it from displacing. It is also important to prevent too much movement across the fracture site so that the callus can form. And so fractures are often treated with immobilization. This can be done with casts, splints, or surgical fixation.
Okay. So this again connects back to this balance between osteoclast and osteoblast activity, and it's going to connect to what we already know about the periosteum. Okay, so let's walk through this again. The first thing that happens after a fracture is bleeding. You guys will learn more about this at AP2, but in general, you form a blood clot, right? These blood vessels in your bone are broken, and therefore blood is allowed to exit your blood vessels. And so through a chain reaction, platelets are going to join together in this positive feedback scenario and essentially start creating this network of proteins and blood cells and platelets that forms, this fibrous network. So first of all, it stops the blood and it starts forming the scaffolding for the formation of that callus. Okay, so very first thing that happens, right, within minutes to the first hours is the formation of the hematoma, which is just a blood clot. Next, over the next few days, you start to form the soft callus, right? And of course, at the same time, there's a lot of inflammation, right? So your body is cleaning up all that damaged tissue, and also, the swelling around that break inherently immobilizes the healing tissue, right? It hurts and like it's soft, and you kind of like, even if it's not immobilized with a splint or something, you naturally don't want to move it. You naturally don't want to damage it even more. And so inflammation is actually a pretty important part of this process and is a natural splint, essentially. But again, the hematoma forms this fibrous network, which is then used by the chondroblasts or the chondrocytes. And so what we can see here in this fractured bone is that the periosteum, thank goodness, is still intact. It is still there. And so what this means is that there are a lot of stem cells, which when they detect that there is damage in the tissue, these stem cells are going to turn on chondroblast genes, which of course forms chondroblasts, which then start secreting cartilage, right? Those proteoglycans trapping water molecules and therefore creating that nice, flexible yet fairly durable cartilaginous callus. Okay? And so again, these cells came from the periosteum. That is a huge, huge importance here. Without the periosteum, right? So if the periosteum is damaged, if it is torn off, or if a burn accompanies a break and it goes that deep, essentially it takes a lot longer to heal, if at all. And so just like with endochondral ossification, we have osteoblasts that can come in and they can start secreting osteoid, which is then ossified within that cartilaginous model, right? So this used to be the soft callus, right? Just cartilage. Now it is the hard callus. It is spongy bone. Just like when we are first building our bones, the osteoblasts just throw down as much bony tissue as they possibly can, as quickly as they can, just so that this person can get back to normal. Only then do we have osteoclasts come along, tunnel, small tunnels into this bone, and so we can get this slow remodeling process. Okay, and so over the next months, over the next years, this callus is going to be remodeled back into what it used to look like before the break. And so here is, or here are a couple x-rays here. Here is a fracture of the fibula. Here is 6 weeks later, so we can see that the fibula is, or has this extra bump where the break used to be. This is not only, just a side effect of how bone heals itself, it also is helpful to have a slightly fatter, maybe, a slightly fatter region of bone here, right? If this spot was weak enough to break before, maybe we want to have a larger bone in this place so that we don't get a second break. And of course, the video talked about the importance of setting bones, as I'm sure you're all familiar with. If there's too much displacement in a bone, like in this radius over here, the bone will heal, probably, but it won't look normal, right? And so oftentimes you have to re-break it, reset it in order to get it back to the original shape. And of course, sometimes this requires surgical efforts. So this here is a femur that has been absolutely shattered and is put back together surgically. Okay. Other types of damage besides just fracturing a bone. An example here is osteoarthritis. So essentially, you can break a bone at any time, but what is also happening over your life is that you are putting so much wear and tear on your joints. Right? So this here happens to be a knee. This is your femur. So this is your thigh bone. This is a tibia. And so if you are, if you are athletic and constantly, playing sports and everything, maybe there's a lot of grinding on your joints in this way. Obesity is linked to, a lot more pressure on this cartilage. And so over time, you get this wearing down of the cartilage, the articular cartilage at the end of your joints. And so remember that cartilage is avascular. It can't heal itself very quickly, if at all. And so if you're continually exerting the stress on your joints, it's just going to get worse and worse over time until, as we can see here, the cartilage can completely wear away and you get this grinding sensation. What can also happen here, right, in addition to the wearing away of the cartilage, is that now there is less space between those bones. And so the edges of the bones might start to rub together as well. So remember earlier in this talk, I told you that the periosteum covers everything on the bone except for where the articular cartilage is. Well, now if the articular cartilage is rubbing away, now maybe the edges of the femur get to rub on the edges of the tibia here, which is essentially scraping at that periosteum. Right? What do you think is telling the periosteum cells, these osteogenic cells, that they need to divide besides abrasion, right? Actual mechanical stress. And so when there's rubbing on those osteogenic cells, the stem cells say, oh my gosh, like there is damage here. I need to replace this bony tissue. The stem cells differentiate into osteoblasts and they start laying down more bony tissue, right? That's what you would do if there was a break, right? If there was a mechanical stress on the periosteum. The problem with this, however, is that we're not supposed to have extra bone here, right? We're not supposed to have osteoblasts laying down more bony tissue on the edges of your poor joints. And so now you have all these little sharp spicules called bone spurs because of that rubbing on the periosteum, because of the damage to the articular cartilage. Okay, and so bone spurs really are just appositional bone growth, right? Stimulated by a lot of stress on your joints. And so this isn't just something that happens in your knees. It can also happen, for example, this is your heel bone, your calcaneus. If you are on your feet all day, if you're wearing lots of high heels, uncomfortable, unsupportive shoes, essentially that puts a lot of stress right here on your calcaneus. And so, rubbing of the periosteum and ultimately getting bone spurs on your heels or heel spurs. Now this is not only particularly uncomfortable because of course you're standing on your feet, now you have bony spurs on your feet, but also there is a lot of connective tissue that runs on the plantar side of your foot, essentially stabilizing all of those bones and muscles of your feet. Now, if you have bone spurs here, where do you think they're going? Besides in the fleshy connective tissue and muscular tissue on the bottom of your foot. And so it is continually rubbing on that and maybe fraying those tissues and causing a lot of inflammation, a lot of pain. And so plantar fasciitis, so itis, inflammation of the fascia on the bottom of your foot can be linked to bone spurs as well. And here in the cervical spine, you can see that there are bone spurs actually on the bodies of the vertebrae. Actually, the images that I gave you guys of the vertebrae models in the lab have bone spurs, right? Also, there's some crazy spurs on the ribs as well. That's not how normal bodies of vertebrae are supposed to look, but the bone clone models that we have are actual castings of real people's bones. So whoever's bones, these models were made from had some pretty rough bone spurs on the bodies of their vertebrae, right? So that can also be associated with the degenerative disc disease, where now there's less space in between the bodies of the vertebrae. And so when you bend your neck, there's a lot of rubbing on those bodies, right? So not only like a pinched nerve kind of thing, but also bone spurs as well. Right, so just another plug for strengthening those stabilizer muscles to hopefully reduce this a little bit. Okay, disease, right? So bone remodeling and an imbalance due to disease. Osteoporosis is the most common bone disease in the United States, often linked to these issues down here. So an inadequate amount of calcium or vitamin D. Estrogen kind of fights bone deposition, right? So it actually decreases the activity of the osteoblasts. And so just being female increases your chances of getting osteoporosis. As you get older, not only are your sex steroids shifting, but also as you get older, as the video said, your repair processes in general just kind of slow down. And as a result, osteoclast activity is still working, but osteoblast activity just can't keep up. Okay. Lack of exercise, right? So if you don't move it, you lose it. Or even, as I said before, if you go into space and you are not needing to withstand the force of gravity, you don't need very strong bones. And so your osteoblasts aren't going to replace that bone as quickly if you're just not using it. And of course, there's a lot of genetic factors. But ultimately, osteoporosis is just porous bones associated with more bone breakdown than bone build up. Okay. And that, oops, we're not going to do open-ended practice because that is not something we need to do for this upcoming exam. Okay, oh my gosh, so much information. Any questions? I know I flew through that quickly. All right. All right, so at this point, that is the end of the lecture information on exam number 2. Your exam number 2 is going to be this Wednesday. I will have Zoom, essentially office hours, to let you guys come in and ask questions. I don't really have anything planned for, you know, an official review, but if you want to come and like hear what we're talking about, by all means, you can join for that. Your lab practical is going to be posted on Blackboard right under the exam 2 resources folder. It's open at 2 today through midnight tomorrow. Same format. I'm pretty much going to have a picture, right, of something like this and say, you know, what is this entire region of the bone? And you'll tell me that's the epiphysis, right? What is this whole region of the bone? You'll tell me the diaphysis. What is this substance inside the medullary cavity, right? So that's the yellow bone marrow, right? All of those terms on the practical 1 terms list are fully fair game for this test. There are reused questions, right? So it is in your best interest to go back and take a look at the practice practical as well as your lab quiz, because you will see some of those questions again. Okay. There are, or this is 50 points. And so there will be 25 stations or questions with 2 structures, bones, whatever each. Some of those questions might have like 2 parts, right? So, you know, for example, going back to this, you know, the cell, like I could point to this and say, what is the cell and what is the cavity, right? So the cell in here would be the osteocyte, the cavity would be the lacuna. And as always, try to use the words exactly as they are on the list. If there are 2 structures, so for example, the ventral root versus the anterior root, see, that is, right, if we're looking at this, sorry, a little difficult to see, but the dorsal root, the ventral root, dorsal root ganglion, spinal nerve. Within the spinal cord, we have the white matter, right, myelinated axons, and we have the gray matter, unmyelinated axons. We have the posterior or dorsal root, the lateral root, or horn, oh my gosh, posterior or dorsal horn, ventral or anterior horn, right? And so on the list, you see that it's like posterior parentheses dorsal horn. Pick one of them and tell me that. Don't give me both. Don't give me lots of crazy slashes, just posterior horn. That's it. And that will allow Blackboard to make the right call, and so you won't have to wait for me to go back in and make a judgment call about whether it's actually right or not. Okay, so that will expedite your grades surely. Okay, let's see. On the spine, remember that there are 3 regions. So if I'm asking for the region of a vertebra, right, it's cervical, it's thoracic, it's lumbar. If I give you a picture of the actual spine, I know the lighting here is not the greatest for you to see this, but you can actually like count down. If I give you a picture that you can, right, see 1, 2, 3, 4, 5, 6, and 7, right? So C7, C6, C5, right? No dashes, no spaces, just the letter C, the number 5. Okay, so from C7, T1, T2, T3, T4, and so on and so forth. Right, so if I'm asking you for the letter number combination, I want T3. Okay? If I'm asking you for the name, for example, right, remember that the vertebra prominence, C7, that's its name and its letter number vertebra indicator. Okay. Let's see. You guys are more than welcome to speak up or ask questions via the chat feature. By all means, questions are great. If there aren't questions, I'm just going to point out a couple more things for you guys. All right. Remember that there are 7 pairs of true ribs, 5 pairs of false ribs. 2 of those ribs are floating and or false ribs, right? So you could tell me floating or false, and that would be okay for these 2. But for the other 3 pairs of false ribs, they're just false, they're not floating, right? And again, you can count, right? 1, 2, 3, 4, 5, 6, and 7 true ribs, 1, 2, 3, 4 false, and 1, 2, or 11 and 12 rather, for floating. Okay. So this, we now know all sorts of things about this. Hooray. Hopefully you guys enjoyed, or hopefully at least got something out of this most recent open-ended video. But muscle cell, right? Remember, this is the motor neuron. That is a cell. This is a Schwann cell, a cell, right? This here is the sarcolemma of the myocyte or myofiber, right? That is a cell, right? On the back here, we see connective tissue. That is the endomysium. We know the endomysium surrounds each and every cell. Okay. Right. The spiderweb looking thing is the sarcoplasmic reticulum. Don't tell me SR, tell me sarcoplasmic reticulum, right? And of course, the triad is all 3 of these. Sarcolemma, the specific part of the sarcolemma that is, essentially covered with receptors for ACH from the motor neuron. That is the squiggly little motor end plate, right? So just a couple reminders that you may or may not see. We'll see. Any questions for me? All right, I'm happy to show you any of the models. I'll hang out for a few more minutes. You can stay behind and, chat more privately if you'd like. If not, I will see you, maybe, it's optional on Wednesday to come to our Zoom meeting. Okay, so if not, I will see you, or at least see your names on a black screen next Monday. Oh, and remember that, a week from today, we are doing our chicken leg dissection. So make sure that you have access to a chicken leg for that. So I'm gonna stop the recording. I'll get this posted on YouTube as soon as I can. I hope you guys all have a lovely, lovely day. Good luck on the practical, and if I don't see you on Wednesday, good luck on the exam as well.